Now we are going to talk about how to prepare a complex medium. जैसा कि आप जानते हैं कि reagents, supplies और equipment culture medium preparation के लिए क्या-क्या चाहिए. Once we have all those things available, we can proceed further to prepare the culture medium. We are going to talk about the complex medium preparation. The complex medium needs yeast extract, peptone, dextrose, and the concentration we require is mentioned here: 1% yeast extract, 2% dextrose, 2% peptone, weight by volume. तो जिस वॉल्यूम को आपने प्रिपेयर करना है उसी हिसाब से हमें रिक्वायर्ड अमाउंट इन ग्राम्स कैलकुलेट करना पड़ेगा. If we want to prepare 500 milliliter medium, we will need 5 grams of yeast extract, 10 grams of peptone, and 10 grams of glucose. All these three materials are going to be dissolved in water in a conical flask of 1 liter volume. The mixing will need the stirring on a magnetic stirrer. जैसा कि आप जानते हैं room temperature पे sometimes the solubility is an issue. For that purpose we have to mix the reagents using magnetic stirrer. Once all the components are dissolved in water, then we are going to split this 500 milliliter into two flasks, which will be labeled as flask A and flask B. Flask A is going to be as it is. No further addition is going to be done in flask A, whereas the flask B will get 5 grams of agar, which will be dispersed and mixed in the broth. इसका जो पर्पस है दो फ्लास्क में स्प्लिट करने का, एक फ्लास्क हम यूज़ करेंगे लिक्विड कल्चर इस्टेब्लिश करने के लिए और दूसरी फ्लास्क से हम एगर प्लेट्स इस्टेब्लिश करेंगे, सो दैट वी कैन स्टडी द लिक्विड कल्चर एट द सेम टाइम वी कैन स्टडी द सॉलिड कल्चर. फ्लास्क ए में से हमने 15 डिफरेंट टेस्ट ट्यूब्स में 5 मिलीलीटर ब्रॉथ शिफ्ट करना है. 5 milliliter of broth is going to be shifted and the test tubes are going to be covered with their caps. At this stage we need to know that the caps should not be tightened very securely because the media still have to go for autoclave and to be sterilized. Now this is the time to place flask A, flask B and the test tubes carrying the culture media, the broth, for autoclave so that it can be sterilized. Autoclave will be done for 20 minutes at 120 or 121 degrees centigrade under 15 to 20 psi. Once the time is over, we have to wait for some time, which is called as venting time, so that the steam which is accumulated in the autoclave should be released and there should not be any accidents or accidental burns to the researcher. After the venting time, open the autoclave and using heat resistant gloves, remove your culture media flasks and test tubes from the autoclave. Let the flask A and the 15 test tubes cool down before securely tighten their caps and storing them for further use. Flask B is going to be used to plate the media into the Petri plates, the sterilized Petri plates, and during shifting the media from flask B to the sterilized Petri plates, I would recommend you to observe maintaining the sterility or the sterilization and don't contaminate your material. Follow all the aseptic methods we already talked about.